Hello everybody. I'm Elena. I was going to say I'm a PhD student, but I guess I'm not anymore. I just had my defense, I guess you would call, in the US 2 days ago. So now I'm a postdoc, but I'm going to be talking essentially about what my PhD has consisted on, which has been looking into sex differences in genetic architecture, and we have been focusing on UK Biobank. So I'm going to talk a little bit about what we've been doing with that. So I guess the first big question is why should we be interested in sex differences? So as you all know, males and females are different. However, these differences expand, just what people might think of in terms of the way that we look or our reproductive function. Differences have been observed in response to medication, as well as the prevalence, incidence of many different diseases from a wide variety of types of disease as well. So, for example, cardiovascular disease is known to be more prevalent. So it's been seen that males are more predisposed to cardiovascular disease than premenopausal women. However, this does change post-menopause. So that's, and a similar thing happens with asthma as well, where boys are more predisposed to asthma, but girls after puberty are more predisposed to asthma. So the reasons behind these differences are not really very well understood. And here I have like a little bit of the hallmarks of the potential contributors to the differences that we're observing across the phenome between males and females. Now, when people think of differences between males and females, they probably automatically think of the sex chromosomes. So the sex chromosomes are the main, I would say, the primary differentiating factor between males and females in humans. You have females with XX chromosomes and males with XY chromosomes, typically. And these do kickstart human differentiation, and thus many times people assume that all differences that we're observing can be are attributable to sex chromosomes. However, it is thought that sex chromosomes, at least the Y chromosome specifically, it is more of a kickstarter. That means that once a person has been fully differentiated, there are many other potential contributors to the differences that we're observing. Beyond just, the difference in actual gene content, other differences could arise. For example, genetic architecture could be different, and that is the focus of this particular study. Now, genetic architecture, if you're not familiar with the term, it's just a fancy term to refer to how genetic variation leads to phenotypic variation. So that can be anything from the effect of alleles to relationships between different genes like epistasis, dominance, also pleiotropy, gene by environment. Basically anything that is genetics leads to phenotype is usually what is referred to by genetic architecture. But beyond your genetics, there are other contributors that could be influencing as well some of these differences. So epigenetic differences have been observed across the methylome between males and females. Genome regulation is also has been widely reported to differ between males and females, with the latest GTEx paper citing around a third of genes presenting sex biases in gene expression between males and females in at least one tissue. So that's a pretty significant number. Then you have endogenous factors. So things like hormones have also been the classic players in understanding sex differences, or, sex differences have been classically attributed to differences in hormones. And then finally, something that people might not really think of is exogenous factors. So because we live in a society with gender roles, it might be that males are more predisposed to enter certain work environments, and those work environments might have particular risks. Same thing for females. And that could ultimately also influence the differences in phenotype that we're observing. However, if we really wanted to simplify it, you could say that there's your genetics and then there's everything else. And that everything else you could say is your environment as defined by your sex. So you have your genetics, and surrounding your genetics, there's differences in epigenetics. There's genome regulation differences. There's a different hormonal content. So all of these things could potentially be interacting with your genetics to lead to some of the differences in phenotype that we're observing. And this is what we are calling a gene by sex interaction. And it's just a way of modeling this. So if you look at the literature, you'll find that there are different ways to study gene by sex interactions, which just make use of traditional quantitative genetics frameworks. So here I've very arbitrarily divided up the ways into the broad view methods and the narrow view methods. Now, with the broad view methods, how I differentiate between the two is essentially what you want to get from this particular method. So with the broad view methods, you want to quantify the relationship of the genome as a whole to the phenotype as a whole. So with a single number, with a single digit, you are looking to quantify how your genetics are potentially associated to your phenotype. Now, an example of a method like this would be heritability. So like the name indicates, heritability is defined as the fraction of phenotypic variation that can be attributed to genetic variation. So essentially how heritable a trait is. If you have a heritability of 1, you could say that the trait that you're looking at is extremely heritable or totally heritable, meaning that it's totally accounted for by your genetics. And 0 would mean the opposite. So heritability, by calculating heritability separately for males and females and comparing, we are able to assess whether there is an equivalent relationship between your genotype and your phenotype in terms of how heritable it is. And the difference in heritability could be a clue into a difference in genetic architecture between the sexes, so the existence of gene by sex. Similarly, there's something called a genetic correlation, which, like the name indicates, is essentially a way of measuring how common the level of genetic control is between either two groups, like males and females, or two traits, which have also popped up in the literature. Again, so a genetic correlation is a single digit that quantifies this relationship between your two groups or your two traits. Genetic correlation of 1, meaning total common genetic control, a genetic correlation of 0. Well, it is a bit more complicated than what I'm just saying. It essentially would mean no commonality in the genetic control. Now, moving from the broad view methods to the narrow view methods, we are moving into the territory of genome-wide association studies, where we are now interested in understanding how each individual genetic variant is associated to the phenotype as opposed to just the genome as a whole. I think genome-wide association studies are probably the most well-known because it's not only quantitative geneticists and animal breeders, it's also bioinformaticians that are familiar with this particular framework, which has gained huge popularity at the start of the century. So similarly to with the heritability and genetic correlations, we are able to run genome-wide association studies separately for males and for females, which then allow us to estimate how each individual genetic variant is associated to the phenotype, which then allows us to postulate whether each individual SNP has the same association to a phenotype. Now, if a genetic variant was to hold a different genetic effect to the phenotype, then we could again assess whether gene by sex is present. Now, if we look at the overall consensus from the literature, especially pre-2017, which is when I started, you will find that differences had been reported, but they weren't very big. Especially what I found is there was a lot of studies focusing in on several types of traits, and those were especially anthropometric or fat distribution traits like height or waist to hip ratio. Diseases, you also saw that some loci had been reported as sex specific, but many times, many people didn't actually compare whether the genetic effect was significantly different. And so I think the overall consensus was that more statistical power was needed to assess gene by sex, as well as to study it for a wider breadth of traits. And then finally, as with all genome-wide association studies, a problem that occurs is that you might find an association, but you don't know what that association entails. You don't really understand what is underlying that phenomenon. So if we can potentially shed light on sex differences, that would also be incredibly helpful in contributing to precision medicine and understanding how different diseases might have different etiologies for males and for females and inform potential treatment plans. So our aims with this study for, first of all, to assess whether gene by sex exists, because at the beginning, we really didn't know beyond what had been published for several traits, assess how, if it exists, what its extent is, how is it very common, not only in the genotype, but across the phenome. And then once that had been established, if it got established, the next step for us was to try to get some information regarding how gene by sex interactions could be arising. So a common hypothesis is that, and a fairly well accepted hypothesis is that gene by sex or genome-wide association study hits on their own. They are essentially mediating gene expression of nearby genes, so acting as something called a cis-EQTL, which essentially means that your genetic variant, which falls within a known coding region, is somehow regulating gene expression of nearby genes, and that regulation is somehow downstream leading to the differences that you're observing in phenotype. So in that context, it didn't seem crazy to think that gene bisex could potentially also be arising. If you were to find SNPs that showed evidence of gene bisex, it wouldn't be crazy to think that maybe they are also regulating gene expression in a differential way between males and females. And then finally, beyond just the effect on the transcriptome, we wanted to see if by using some bioinformatics pipeline, we could shed light on mechanisms for gene bisex. Is it possible that all of these SD SNPs or these sexually dimorphic SNPs, is it possible that all of these are falling within a particular pathway? Is it possible that they're all within binding sites for hormone response elements, and so on and so forth? So our main data, like the title indicates, was UK Biobank. I think most people know what UK Biobank is, but if you don't know, UK Biobank is essentially a population cohort from the UK, like the name indicates, with around 500,000 individuals. Now, the individuals that we're focusing on here is around the 450,000 white European individuals. There is a bias with more females than males. And after QC, we have 530 different traits. Now, these traits are both non-binary and binary. So the non-binary are continuous traits most of the time. So things like height, for example, whilst the binary traits are case-controlled traits like diseases. So whether or not you have had a particular disease. And then finally, after QC, we have around 9 million genetic variants left as well. Now, whilst the methodology is quite varied, truly at the heart of everything is something called a linear mix model, here specifically using the software Dissect. Now, like I mentioned, going back to the broad view and the narrow view methods, what these models allow us to do is estimate the relationship of both the genome as a whole to the phenotype, but also the relationship between each individual genetic variant and the phenotype. So because we know that phenotypic variation can be divided up into variation accounted for by your genetics and your environment, using linear mix models, we are able to obtain genomic parameters, which is a fancy term for essentially these variance components that inform us on the relationship or the amount of variation within a phenotype that can be accounted again by your genetics and your environment, but also allow us to estimate something called a genetic effect, which is essentially, the effect that each individual genetic variant has on the phenotype. So this is just the formal name for what you get from a genome-wide association study. Now, by doing this separately for males and for females, this then allows us to compare and assess whether these SNPs or whether these genomic parameters are significantly different between the sexes. So we did this separately for males and females across the 530 traits that we are considering here. So going back to the broad view methods, the first question that we wanted to answer is there a difference in heritability between males and females? A recap, heritability is the fraction of variation of the trait that can be explained by the genotype. What we found is that 30 of the 446 traits presented a significant difference in heritability, whilst for the non-binary traits, it was even more, 41 of 84 had a significant difference in heritability. Now, if you're not familiar with the equation for heritability, it essentially is like this, where you have the genetic variation of a phenotype, or the genetic variance. The A is for additive genetic variance. So that's in the numerator, and the denominator you have both that the genomic additive variance as well as the environmental variance, which means that a difference in heritability between males and females could potentially arise because there is a difference in environmental variance between males and females. So because we are interested in the genetic component, we've further assessed whether these differences in heritability were being driven by genetic variance differences or environmental variance differences. So here I am showing just for a couple, just to showcase what we mean by this, just a couple of traits and as well as their log to male to female variance ratio. You can see that all of them have a ratio of variance for the genetic variance that is larger than the residual environmental variance. And we found that this was the case for the majority of the traits considered. So we could conclude that in essence, the majority of the differences in heritability that we were observing were being driven by differences in genetic control as opposed to just differences in environment between males and females. So moving from heritability to genetic correlations, we were interested in assessing whether we could find genetic correlations that significantly differed from one, because as I mentioned before, genetic correlations are essentially the level of common genetic control between two groups. So a genetic correlation of one would mean no difference in genetic control, essentially. And a difference from one would indicate that there is a level of heterogeneity in genetic control for a given trait. So here we use something called LD score regression, which is a bit more limited in regards to for the binary traits, where they recommend that you have at least 5,000 cases. So that did reduce the number of traits that we considered for this part of the analysis. But even still, we found that 11 of those remaining 77 binary traits had a genetic correlation that significantly differed from one. Whilst for the non-binary, it was a very big number. It was 58 of the 83 had a genetic correlation that significantly differed from one. Now, something quite important, I think, is that even though these genetic correlations significantly differed from one, and here I have a histogram showcasing the value of the genetic correlation and the number of traits that presented said value here represented in the x-axis, even though they significantly differed from one, the majority did lie close to one, meaning that differences were not huge. So there is a level of heterogeneity in genetic control, but it's not very big. There were some traits that had a genetic correlation that significantly differed from one and that were further away from one. So things like disorders of urinary tract, hernia, eczema, gastric reflux, and then waist-to ratio, which I'm going to be talking about a little bit more because it is the trait that has been discussed a lot in the literature regarding gene by sex interactions. We also compared our genetic correlation estimate as a sanity check across other studies. And there were several studies that had looked at genetic correlations between males and females for several anthropometric or fat distribution traits like waist-to ratio, body fat percentage, BMI, waist circumference, hip circumference, and height. And here in pink, you can see our result with the 95 confidence interval also plotted. And in blue, the results from other studies. And you can see that they do coincide very well. All the confidence intervals are within each other, overlap somehow. So that was a good sanity check for us as well. Okay, so having established gene by sex or having assessed it in terms of heritability and genetic correlations, we next moved on to what I think is potentially the most interesting part, which is the sex stratified genome association study. So what we did is we compared for each individual genetic variant across the genome, whether its genetic effect was significantly different between the sexes. Excuse me. So these SNPs that we found to be significantly different between males and females, we termed sexually different variants or sexually different SNPs or SD SNPs. So I'm going to be referring to them as SD SNPs from now on. And we found that 61 of the 84 non-binary traits considered in the study had at least one SD SNP. Whilst for the binary traits, it was 42 of the 446. Now, bear in mind that here with UK Biobank, we are more limited with statistical power when it comes to the binary traits, because UK Biobank is mainly a healthy population. So we don't have huge numbers of cases for certain diseases, as opposed to consortia that are honing in on a particular disease. However, we still found pretty interesting things. Here I have in the two tables below the non-binary and binary, I have the traits that presented the largest number of SD SNPs after LD clumping, so independent hits. And you see waist-to ratio by far is the trait that presented the largest number of independent SD SNPs, but you still see some other anthropometric traits like height. And with the binary traits, very interestingly, we find that ankylosing spondylitis, gout, hypothyroidism are showing up. And these diseases have been reported to present differently between the sexes. So here I have a Manhattan plot for the results of waist circumference ratio. If you're not familiar with that Manhattan plot, essentially on the x-axis, we have the genomic position. So you have chromosome 1, chromosome 2, and then within that, you kind of linearly go along the chromosome. Each point represents a genetic variant, and the height is the minus log 10 p-value of association to a particular trait under study. Now, here in this case, the p-value is not necessarily that. It's the p-value corresponding to the t-test that we used to compare the genetic effect between males and females. So the higher up the point is, you could say, the more evidence we have of sexual dimorphism between the sexes. So all of these peaks correspond to to loci in the genome that present evidence of sex differences in genetic architecture or in genetic effect, essentially. Like I said, waist-hip circumference ratio was the trait that presented the largest amount. In total, here we are showing, in total here, we have 100 independent loci that showed sex differences. And also has a sanity check recently, or fairly recently, in 2019, Pollitt and collaborators, they published a meta-analysis using both GIANT and UK Biobank data. And what we did is we took their summary statistics because they do actually do a comparison themselves, but to make it as fair a comparison as possible, we took their GWAS summary statistics for males and for females for waist-hip circumference ratio. We ran them through our t-test and we compared, and here you can see that the p-values correlate very, very nicely with our own results, which again, was a good sanity check. Okay, so this is a portion where we have now seen that gene by sex seems to be present. We've assessed this with heritability, genetic correlations, and genome-wide association studies. Now, this is great, but what can we actually do with this? How is this useful? Because it is, in an ideal world, this would inform us on the biology of many different types of diseases, but it's truly not that simple to get biological insights from genome-wide association studies. There are, however, other more immediate applications of genome-wide association studies. Now, nowadays, genome-wide association studies are used for phenotype prediction. So once you've run a model and you've obtained estimates of genetic effects, you are then able to use your estimates and input the genotypes of new individuals and assess or try to predict the phenotype of these new individuals. Now, nowadays, the majority of these prediction analyses are done using summary statistics, GWAS summary statistics stemming from a mixed population cohort. So a cohort that has used both males and females, sex combined, sex agnostic, whatever you want to call it. So this is great. However, if there are differences between the sexes in genetic architecture, it could be that a sex-specific model might be better for the prediction of phenotypes than a sex-agnostic model. So this is something that we wanted to explore. And another important consequence of the existence of gene by sex is the potential existence of masking. Now, what we mean by masking is that it could be that a genetic variant has a genetic effect that is interesting in one of the sexes, be that because this particular genetic variant is binding to a sex-specific factor, such as a hormone, in just one of the sexes and not the other, and thus this genetic variant is only relevant to a phenotype in males or females. So by combining the data of both males and females, you could ultimately get a non-significant signal. And in an even more extreme case, you could say that a genetic variant is interesting in both of the sexes, but it presents a negative effect in one sex, a positive effect in the other. Thus, when you combine both in a genome-wide association study, you could ultimately end up with a non-significant effect or a zero genetic effect. So both of these things are things that could affect us in the more immediate future, and so we really wanted to explore these things. So starting with the improvement in prediction, what we did is we took the unrelated white British from UK Biobank, which is around 344,000 individuals, and we divided those up into the training and to a testing data set. Now with the training, what we did is recalculated genetic effects. And you might be wondering why, when we had already calculated our genetic effects using linear mixed models, which are ultimately the gold standard. Now here, what our reasoning was that we wanted to match sample sizes. So in our linear mixed models, we had an unequal number of males and females in each model. And if we were to compare it with a sex agnostic model, that would even be more unfair, since the sex agnostic model has essentially twice the sample size as sex stratified efforts. So what we did is we took the 300,000 and we ran a model for just males with 150,000 males. We run a model with just females with 150,000 females. And then for our sex combined model, we ran in 75,000 males and 75,000 females within the same model. And so we reobtained our genetic effects, and that is what we used for our testing data set. And for our testing data set to assess an improvement in prediction, what we did is we obtained polygenic scores, which is just a fancy term for a phenotype prediction. And they are very easy to calculate. Essentially, all you do is you take the genotype of the individual for which you want to predict the phenotype, you just input in their genotype values, and you multiply each individual genetic variant by your estimated genetic effect from your models or your genome association studies. And then you just sum all of that up. So to compare, we did this in 3 different ways. We obtained the same polygenic score, by which if you're female, then we would use your genetic effect as estimated in the female specific model. If you're a male, then we would use the genetic effect estimated in the male specific model. Then we obtained the opposite polygenic score, which would be the opposite of what I just explained. If you're a female, then we would use the genetic effects obtained in a male specific model. If you're a male, the genetic effects obtained in a female specific model. And then finally, the agnostic polygenic score, which is truly what we want to compare to, which is independently of your sex, we're going to be using a genetic effect that has been estimated using a genome association study that has accounted for both males and for females. So it hasn't stratified. So we did this for all traits with at least 10 SD SNPs. And sadly, we didn't find an increase or a statistically significant increase in prediction. Now, to leave it on a more optimistic note, however, here I have the results of, here on the y-axis, I have the correlation between the prediction and the actual phenotype value. Now, keep in mind that these predictions were done using only SD SNPs. So it could be that some of these only used 10 SD SNPs for the prediction. Hence the correlation being very low. It's not the greatest prediction, but here what we're trying to do is compare how these SD SNPs can fare in comparison to between the same and the agnostic prediction. So you can see that the difference between the agnostic polygenic score shown here in the middle and the difference with the same polygenic score shown on the right-hand side is not very big. However, there was, for all of the traits that we considered, which are shown here, all of these had at least 10 SD SNPs, you can see that there is a slight increase in the actual magnitude of the correlation. So there did seem to be a trend of increased prediction accuracy. Now, it is very important that we found parallels to the missing heritability problem in that these SD SNPs are actually not accounting for a large proportion of the heritability that we found to be different between the sexes, meaning that it's potentially, as larger sample sizes become available, we will find more SD SNPs. And together, if we use these to obtain polygenic scores, these will ultimately potentially be more accurate in terms when compared to the agnostic. But of course, this is yet to be seen. So moving on from the prediction and into the masking, which is, again, another very important thing if it is happening. Again, kind of a recap, what our thinking was with this. It could be, again, that a genetic variant is interesting in just one of the sexes, but not the other. Thus, that when combining both, you ultimately get a non-significant signal in a genome association study. So to assess masking, what we did is we looked for genetic variants that were significant in a sex-specific model, but were not significant in a sex-combined model. So a genetic variant that could ultimately just be significant for males, a significant for females, but ultimately not significant in a sex-combined model. So this is what we are calling masking, and we found a very large number of traits that had at least one masked SNP. So 78 of the 84 non-binary had at least one masked SNP, whilst the binary, it was 98 of 446. So that's a fairly large number. Now, bear in mind that we need to replicate this in other cohorts. However, it is quite alarming that potentially there are things that we are missing, and this masking has also been reported in other biological levels. So if you look at the latest GTEx paper, they also mention that when trying to co-localize sex-specific EQ-tails, you found co-localizations that were not found when using sex-combined EQ-tails. So this could be something that is happening, well beyond the realms of just your genetics. And it does make an important case for us doing not only sex-combined models, but also sex-stratified models to make sure that we're not missing anything. Okay, so we've now gone over gene by sex. We know it exists to a certain degree. We've looked at its potential consequences in regards to prediction as well as potential masking. So I guess the final question is, why is gene bisex occurring? What characteristics do these SD SNPs have in common? Do they fall within a particular motif? Are they binding a particular protein? What is leading to these sex differential associations? So to try and answer some of these questions, we did a gene set enrichment analysis. Now with gene set enrichment, essentially what the reasoning behind gene set enrichment is that because a lot of pathways are well characterized nowadays and they are available online across many different repositories, we know the genes that are contributing in many different pathways that have a particular function as per Go ontology. We also know, thanks to ChIP-seq analyses, the binding sites of particular proteins. So all of these are many times summarized in something called a gene set, which is just a list of genes that have a shared commonality. So we wanted to see whether our list of genes, which I'll explain in a second, but essentially if you have a list of genes of interest and you want to see if these genes are enriched for a particular characteristic, you can do gene set enrichment to see if those genes are more enriched in a particular gene set than a random set of genes of the same size. So to do this, because we do have GWAS data, which is in the SNP form, so we have the results of sexual dimorphism testing for SNPs, the first thing that we wanted to do is take our SNP data, translate it to gene data. So essentially, to do that is quite simple. We use something called MAGMA. What MAGMA does is it maps SNPs to a nearby gene. It takes the data of SNPs and it aggregates it for the nearby genes. So in the end, we are no longer talking about sexually different SNPs, but we're talking about sexually different genes. These genes having a p-value that is essentially a combination of the p-values of the nearby SNPs. And further to that, what we did is we separated them into genes that had a larger effect in females and those that had a larger effect in males. Now, our reasoning for this is that instead of just chucking in all genes that seem to have some evidence of being sexually differentiated, we wanted to see if they are sexually differentiated, is there a particular like female specific pathway that is enriched for those that have a larger effect in females versus a male specific pathway that is enriched for those that have a larger effect in males. So once we have our genes or lists of genes, we moved on to the actual pathway enrichment, for which we used FUMA, which is a web-based application or a tool essentially, which helps you do the gene set enrichment, and it looks for enrichment across a huge variety of different categories or different gene sets. So we did this for a total of 10 traits, which isn't very much. We focused on those that had the largest amount of sexually dimorphic SNPs or genes. However, we found a lot of heterogeneity. So sadly, we didn't find any huge commonality. We didn't find a very easy explanation for SD SNPs essentially. Now, I guess it was naive on our part to think we would, because biology is never that simple. But yeah, we found essentially no size fits all. We also saw no clear connection to sex hormone regulation, meaning that we didn't see, beyond a couple traits where you did see some sets that did have to do with hormone regulation and so on, we didn't really see a common pattern where all of these genes seem to be enriched in a particular sex hormone pathway or sex hormone gene set. Yeah, okay, I see that Lauren has raised her hand.
Hi, thanks, Elena. Yeah, this is fascinating. I'm wondering, it's just such important work. I'm wondering, because it seems like so much of this would be linked to hormonal regulation.
Yeah.
Especially looking at the GWAS traits that were differential, like nip ratio and things like that. Do you think part of the reason why you might not be finding gene sets that are linked to hormonal regulation is because the phenotype that you're using in those GWAS are in a sense, a downstream effect of this kind of endophenotype, which is hormonal regulation? Like if you could do a GWAS of hormonal regulation, however you would measure that, do you think it would be easier to kind of unearth those patterns?
I definitely think that that is a possibility. That's why I think, not here, but in the conclusions, I wrote down direct hormonal regulation, because of course biology is extremely complicated. So it could be that these genes are somehow downstream from those, the hormone might be acting upstream, and then somehow all of that is trickling down, and that is what we are detecting. We don't know yet. I think there's so much more work left to be done. And I don't think that hormones are not a part of it, but I think it's potentially If we just focus on that, we might be shooting ourselves in the foot because it might be more complicated than that. If you look at the GTEx papers as well, they also noted that the genes that had differential gene expression, I think only about a third of them or a proportion of them also were nearby sex hormone receptor binding sites. So also those couldn't just be explained via hormone regulation or direct hormone regulation. So I think we will see. There's so much more work left to be done. This is just an initial, and I do think that this gene set enrichment analysis that we did was quite superficial. I think there's a lot of things that could be improved. So I guess, yeah, it's definitely, I think you're right. We will see if this is just a downstream consequence of hormone regulation.
I'm guessing this is probably a no because it's maybe not something that's super easy to measure, but has anyone done like a GWAS of like testosterone levels or?
Yeah,
like
yes.
That...
Yeah, they have actually. And I think I recommend one by Jonathan Pritchard. It's an eLife paper. It's from this year, actually. It's more, the paper itself was more so focused to try to connect to the omnigenic model. But in itself, they found very cool biology for testosterone. And they were able to link it to testosterone regulation differences between males and females. So I recommend that as reading. Now, how we could link that to our study, I would have to have a think because I definitely think that it would be very interesting to see how hormone GWAS could inform SD-SNP detection and so on. But I would have to have a little bit more of a think about how we could do that. But yes.
Okay, thank you.
Like, both this year and last year have been pretty good in terms of, there's several papers that have come out with GWAS for different hormones as well as other biomarkers. I can send them to you later. I don't know them like at the top of my head, but.
Okay, thanks, Elena. Yeah, that's great.
Yeah.
Thank you.
Okay. So finally, the last thing that I wanted to cover is, having looked at gene set enrichment analyses, we wanted to also see if we could connect it to gene expression regulation. So like I mentioned at the very beginning, gene expression regulation has been thought of as or gene expression or transcripts have been thought of as they're a very accepted endophenotype by which potentially variants in non-coding regions could be acting and contributing to variation in phenotype. Essentially, especially in the postulations of the omnigenic model, where they say that all genetic variants are essentially cis EQTLs for a nearby gene. This led us to believe that it's a worthwhile endeavor to assess whether SD-SNPs are acting as sex-biased EQTLs. So what we mean by this is the following. Here we have a little diagram with a potential SD-SNP, a hypothetical SD-SNP, and a nearby gene. So what we wanted to test is potentially these SD-SNPs could be binding sex-dependent factors, and this could thus lead to a difference in gene expression regulation based on your sex. It could be because a change in allele could affect the binding site of the sex-dependent factor only present in one sex or more prevalent in one sex. Thus, for this particular gene, which seems to have a sex-dependent factor in males, you would see that a change in allele for a particular SD-SNP could lead to a difference in gene expression regulation based on your sex. So that is essentially what people are terming a sex-biased EQTL. And now what we wanted to do is thus test whether SD-SNPs were doing that to nearby genes. So we took a 1 megabase window, and for each SD-SNP, we looked for nearby genes. And so we obtained SD-SNP gene pairs based on that. And to test this, what we used is the GTEx cohort, especially the V6B, specifically the V6B cohort. Now, if you're not familiar with the GTEx cohort, it's very different to UK Biobank, but it's extremely valuable in that, as opposed to many other cohorts that analyze gene expression and its connection to the genotype, GTEx tries to do so from a whole body point of view, in that they are focusing in on a total of 39 tissues, whilst other analyses usually just focus on the most readily available tissue, which is blood. However, blood can be quite limiting, because it could be that many of these traits are not acting via the regulation of gene expression in blood, because blood is not that relevant to a particular trait. Could be, for example, for cardiovascular disease, you might be more interested in looking at heart tissue or so on. So in this particular case, because we are doing a cross-phenotype study, we thought that GTEx would be perfect because it does allow us to have that level of exploration beyond just the blood tissue. However, there is a big limitation to GTEx, and that is its sample size. So in total, we only have 450 individuals. And not only that, but if you actually look at the samples that you have per tissue, you go down to 72, but you have up to 361. So regardless of that, we went ahead with our sex-biased EQTL analysis. And if you're not aware of what an EQTL analysis, don't worry. If you understand what a genome-wide association study is, then and if you understand what a linear model is, then you understand an EQTL analysis. So here I have a little equation where here, as opposed to a GWAS, and a GWAS, Y would be the phenotype value. Well, here, Y is our gene expression value. So what we're trying to see is how gene expression is associated, or how both your genetics as well as your genetics and sex, so an interaction term of SNP times sex, could potentially be influencing gene expression. So if we were to find an interaction term that was significantly different from 0, then we could establish the presence of a sex-biased EQTL. So we ran these models for all tissues, as well as for all of the SD-SNP gene pairs. And what we found was the following. So we found a total of sex-biased EQTLs. Overall, larger sample sizes are needed. So do take this with a grain of salt, especially because if you look at the literature, people have found that it's really, really difficult to replicate these sex-biased EQTLs. And a potential reason for that is that there are differences in cell composition between males and females that could be leading to the detection of some of these sex-biased EQTLs. However, I am going to talk about one of the most interesting ones that we found, which again, bear in mind that it does need replication, hence my writing story time, because we truly don't know if this is true or not. But I will talk about it because I think it also highlights the hypothesis generating potential that these studies have, as opposed to previous studies where, in the past where people set out to answer a particular hypothesis. These analyses allow us to they give us candidate sites to look for more evidence of gene bisex. Okay, so for this particular SNP that we found, I'm not gonna read the name because it's just a bunch of numbers, but this SNP, which fell close to the gene MICA or MICA, which is in chromosome 6, we found that this SNP was regulating gene expression of this gene in a sexually different way. Here you can see that we are quite limited in regards to some of the genotypes. So here you see the homozygote A's. There's far less samples than you have for the actually homozygote G. So do bear in mind, but still we found a significant interaction term. Here in pink you have males and in blue you have females. So each point represents an individual, and the point where they are would be the genotype that they had, as well as their gene expression levels in the y-axis. And so I thought this was particularly interesting because this SNP has been we found to be an SD SNP for ankylosing spondylitis, meaning that this SNP has been we found to have a genetic effect that differed significantly between the sexes for ankylosing spondylitis. Now, I'm not an expert in any means about ankylosing spondylitis, but ankylosing spondylitis is essentially a disease where you have joint and back pain, the result of inflammation. Which is, again, why I found this SD SNP interesting, because we didn't really find this EQTL in a random tissue. We found it to be an EQTL in musculoskeletal tissue, which seemed like a fairly relevant tissue as well to this particular disease. And not only that, but MICA, which is the gene which we found this SNP was acting as a sex-biased EQTL, has also been linked to ankylosing spondylitis. So naively, we could say that this SNP could be regulating this particular gene in a sex-biased way, thus leading to a difference in susceptibility, difference in association between the sexes to this particular disease. Now, to add more to the story, we also found that this SNP has been shown to bind in ChIP-seq analyses to FOXA1. Now, FOXA1 is a protein that also modulates the transcriptional activity of nuclear hormone receptors. So what it does is estrogen comes in, and from my understanding, estrogen comes in and it binds to estrogen binding receptors, and then that binds to FOXA1, and that allows then to regulate the transcription of the gene that is nearby. So, I'm not exactly sure how this could work, But naively, you could say that FOXA1 is somehow associated to hormone levels, which we know differ between the sexes. Thus, you could postulate that this could also be contributing to this chain I've drawn here, ultimately leading to a difference in susceptibility to ankylosing spondylitis. So like I mentioned, this is just a story, this is just a hypothesis, but again, it does highlight how when larger sample sizes become available, once we resolve the cell composition issues, once single cell data becomes available, the potential that these studies have to give us some insight into potential differences between the sexes. So I guess we've arrived at the conclusions, and I wanted to leave you with the take-home messages from everything I've just gone over. The main, I think one of the most important take-home messages is that we did find small but quite widespread sex differences in genetic architecture. We will get even more evidence of this for binary traits with consortia honing in on particular diseases, because like I mentioned, we were quite limited in regards to some diseases. So for studies focusing on cardiovascular disease, for studies focusing on other diseases which have far larger sample sizes, I hope that they take the time to also not only do genome-wide association studies for males and females together, but also sex stratify, see if they can find some evidence of sexual dimorphism. In regards to improvement in trait prediction, we will see with larger sample sizes, again, it's still yet to be seen. We found indications that it could be improved, but larger sample sizes are needed. Masking is potentially where I think ultimately there is more evidence for urgent action, in that it does make a good case for sex-aware frameworks. We shouldn't just chuck our data in and assume there is no difference between the groups that we are including in our data. Beyond direct hormonal regulation, we don't know. Again, far more studies are needed to truly understand what underlies SD SNPs. And again, with gene expression, time will tell. We need larger sample sizes and we need single cell data. And then the final slide, I swear, is that much is left to do. So I wanted to leave you with kind of where I think the study of gene by sex might go next, and where we should go next, essentially, to get more information about the spread and get better understanding of it. So again, like I mentioned, larger sample sizes and sex awareness will be essential in that regard. We've also only talked about common genetic variants, because that is what we use in microarray chips at the moment. Now UK Biobank is releasing exome sequencing, whole genome sequencing. So that will potentially give us also insights into rare variants and what role they are playing in all of this. Sex chromosome methods. So you will find that the majority of genome-wide association studies only look into autosomal genetic variants. However, the sex chromosomes are many times ignored. In this particular study, we did look at the X chromosome, but we only looked at genotyped variants. Imputed genetic variants for the X chromosome were not available when we took our data and we ran our models, but now that is a possibility. So more information can potentially be gathered from sex chromosomes, especially now in recent years where imputation algorithms have been created for X chromosomes. Another important point is that here we're only looking at white Europeans, meaning that you can't really translate these findings to the whole population. We need larger ancestry studies. We need to understand gene by sex, and it will be invaluable, because also doing it across ancestries, we will be able to find map SNPs with gene by sex, and it will be extremely useful in not only understanding how gene by sex occurs in a huge variety of ancestries and make this truly translatable to the clinic across many different populations, but also it'll help us find map gene by sex that seems to be carried over across many different ancestries. Another important point is that gene by sex is very likely a function of time. So like I mentioned in the introduction, you have things like cardiovascular disease, which is more prevalent in males pre-menopause. Post-menopause, it's more prevalent in females. So because hormones are very likely to have a play in this, and hormones change throughout lifetime, it's not crazy to think that gene by sex will also change throughout lifetime. Because UK Biobank is older, this means that what we're showing you here is likely a representation of gene by sex in an older population. We don't really know how that translates over to younger population. As I mentioned, single cell will be essential, especially with gene expression, with methylomics, all of that stuff, which leads me to multi-omics. We still don't know how all of this translates to other biological levels. And finally, here, we didn't really do much causal inference. We don't know if these SNPs are causally associated to phenotypes. So that is another line of inquiry that we should go into. So with that, I leave you. I thank my supervisors, Albert and James, and all my lab team for, without who this wouldn't have been possible. And if you're interested in more stuff, this is the paper, and all the summary statistics are available for you to play around with as well. So I think now I can take questions. Should I be the one to call people, or are you gonna?
I'll do that, Elena.
Perfect.
So I'll ask everyone to raise their hand, and we'll queue up, and we'll have Philip go first.
Okay.
Yeah, so this is super cool, really impressive project. So thanks so much for presenting this. And actually, so this last slide answers my first question, because I was wondering if you've actually written all of that down somewhere. So this is great.
I can send over my thesis where it goes in more detail, but
I actually would love to have a look at that.
Yeah.
It would be great. Yes.
Yeah.
So I have two other questions. So one thing that I was wondering about, so arguably the largest genetic differences between males and females is whether they have a Y chromosome or not, right? So we haven't really talked about that. Do you have any ideas about how we may actually integrate, the Y chromosome in understanding genetic differences between males and females?
Yeah, so the Y chromosome is a bit tricky, mostly because it doesn't really undergo recombination. So there's not really any imputation algorithms. There's, from my understanding, there's no genome-wide associations that have been found for the Y chromosome. Only haplotypes have been found to have associations. So truly, I think methodologically, there's some issues that need to be overcome before we can use the Y chromosome. Yeah, I think at one point I was asked whether we could consider gene bisex as just epistasis between genetic variants from the Y chromosome and the rest of the genome. And while I do think that's important, I do think it's also important to think that the Y chromosome, whilst important, I mean, it's a Kickstarter for sexual differentiation, but it doesn't mean that it's active throughout your lifetime and that it's doing something throughout your lifetime. So I'm not entirely sure just how relevant the Y chromosome might be. I mean, it's naively you could say that, it is a huge difference. It is what kickstarts sexual differentiation. Now, the role that it plays afterwards, I'm not exactly sure, and how we can do it, I'm not exactly sure. I would have to think about it. But I think there is a way to go before we need the technology to truly understand how we could use Y chromosome variation in association studies. Like I said, at the moment, only haplotypes have been found to have associations within the Y chromosome. So first, we would have to potentially look into that. But that is a good line of questioning as well.
Yeah. So I mean, maybe one first step would be thinking about how we could quantify the heritability of phenotypes among males that can be attributed to the Y chromosome.
Yeah.
So that would give us a first hint of how important that actually is.
Yeah.
But yeah, I totally agree. This is a total wormhole, and we've stayed away from the Y until now for all the reasons that you mentioned. And another question that I had was regarding polygenic prediction of phenotypes. So maybe I misunderstood something, but it sounded like you're suggesting that by running sex-differentiated GWAS, we may end up with polygenic scores that are more predictive of phenotypes.
Yes.
But the obvious problem there is that if you do a sex-stratified GWAS, you're basically throwing away half of your sample size, right?
Yeah, exactly. Yes. So that is a very important point, because we need to, I mean, the differences have to be large enough where the advantages of those differences have to surpass the increase in statistical power. So from what we did, I don't think we're there yet, because I'm pretty sure before we matched sample sizes, the agnostic polygenic score worked slightly better than the sex specific. So like I mentioned, take that with a grain of salt, and that more work needs to be done. I think more SD SNPs need to be found if you were to use those for prediction and for that to surpass the agnostic. But like you said, at the moment, I don't think it's worth it, because the larger sample sizes combining males and females will likely give you a better prediction than half the sample size using your own sex.
Yeah. Okay. Thanks so much.
Yeah.
Thanks. Q, could you go ahead?
Yes. I just want to say that this might be the most well-designed slides I've ever seen in a long time. All the color choices, it's just perfect. I really appreciate that.
I changed the Nature Genetics color here just so it would match. It's actually green in real life, but
yeah, you really appreciate these details. I think it helps a lot. So thank you for that. I have maybe two questions. The first one is related to Philip's question. I was going to ask about X chromosome because I see that you did include that in your analysis.
Yeah.
I wonder if you can comment on that. I don't think you mentioned very much about it in your presentation,
no.
Basically, if X account for phenotypic differences between males and females, that will be my first question.
Yeah, so we did include the genotype. So we used the UK Biobank 2017 release, which included genotyped variants from the X chromosome, but they had yet to impute the genetic variants within the X chromosome, meaning that we only had 17,000 variants, which in genome-wide association terms is not great because LD blocks are quite small. Therefore, you don't have we didn't have as many associations. In fact, we found no associations in binary traits. We only found them for non-binary traits. Yeah, I mean, we didn't really find very much, but I guess my point is that we need more work in regards to the X chromosome. So although we did look at the X chromosome, we didn't look at it in a lot of depth because of the limiting data that we had. And also, like with the Y chromosome, the X chromosome is a bit tricky because here we are assuming that females have 2 copies when truly one of them is inactivated and we don't know which one it is. So I think, again, as with the Y chromosome, methodologically, we still have some ways to go to truly understand how variation in the X chromosome affects phenotypes, because, like I mentioned, for females, we don't really know which is the variant that is being expressed or that is influencing expression or that is being transcribed.
Yes, just a very quick follow-up. So in female-specific analysis, you just use standard GWAS model for X? Okay.
Yeah.
I see. So my second question is about a formula you had. So I wonder if you mind going back to the slides where you show the Manhattan plot for weight-skip ratio.
Yeah, of course.
So here you have a test statistics to identify these SD SNPs. I think that's how you call them.
Yeah.
I wonder if you can comment on the third term on the denominator, which is a little bit odd.
Yeah, it's the covariance essentially between males and females. So there's many studies that don't include this term. But some studies. So this is the correlation.
First of all, I assume there should be a square root somewhere, right? But the term is still.
I think I may have forgotten the square root now that you mention it. That is my bad.
I just noticed it actually, but
yeah, I didn't notice either. That's a good catch. Yeah, I'm not sure if this little quotation mark is an indication that something in the formatting has gone wrong. But anyway, yeah, the third term is supposedly representing the covariance in genetic effects between males and females. And I'm not a statistician, so I might butcher this. But essentially, this term, you can remove it when you assume there's no covariance. But in this case, so this is the Spearman rank correlation, I believe, in genetic effects between males and females for a given phenotype. Or is it? It's better explained in the paper than what I'm saying at the moment. But essentially, there are studies that compare genetic effects and they don't include this term. However, because R was not, we calculated R and we found that this R was not close to 0 for some phenotypes, it didn't seem appropriate to remove the term. So we included it. I can't go, I'm afraid, far more into detail because I'm afraid I might butcher the statistics behind it.
I'm sorry, I'm still a little bit confused because when you talk about some sort of correlation, you're thinking of two things measured on the same set of samples, right?
Yes.
But if you're talking about the correlation between males and females, these are different.
I'm talking about different, the genetic effects. So I'm pretty sure the correlation is representing the correlation between the genetic effects. For a given trait, you have your SNPs in males and your SNPs in females. I'm pretty sure that is the... Okay.
I see your point, but so this is not exactly the same, but it's pretty similar to the idea of genetic correlation. Then,
yeah,
right?
it's similar. But again, don't hold me to that because I might be wrong. I don't remember at the moment how that R was exactly calculated. But the take-home message is that it supposedly captures some sort of correlation between males and females that we should be taking into account when comparing because it's not totally independent. It's not, they're not total, yeah.
Yeah, it's, we don't have to get into all the detail, and we have a one-on-one session later, so we can follow up on this. But
yeah,
the very short version is, you would think that, you know, the male-specific GWAS and female-specific GWAS are conducted on different people.
Yeah, they are. Yeah.
So then the test statistics in sex stratified analysis are actually independent. So when you calculate this difference between two sets of summary sets, they are supposed to be statistically independent, right? But this is why I got a little bit confused.
Yeah, I can see why it's confusing, definitely.
Yeah, and a very quick follow-up, it's just suppose you actually run a SNP by sex interaction test in a linear model. It's very basic linear model with interaction effect. How would that differ from this summary stats-based version you're calculating?
Yeah. So we actually, one of our sanity checks was actually running a linear model with a gene by sex interaction to ensure that it didn't really differ very much from our own study where we ran sex stratified and then compared. From speaking to someone that is way smarter than me in regards to maths, they said that mathematically it's quite comparable. I think the only real advantage is that you include less, you have to estimate less parameters, because instead of running two different models, you're running a single model and then including an interaction term. Not sure that supposedly might increase your power, but truly with the amount of samples that we have, it's quite negligible. And with the simulations that we ran, where we ran the models using a gene by sex interaction term, we found very similar results. So I wouldn't expect it to change very much.
Thank you. Yeah, this is great.
Yeah.
Any other questions from the group? Lauren.
I had another question too, but I didn't want to like jump in if any students had questions or wanted to speak up first. I had a question about the single cell technology.
Okay.
We give some of us an intuition a little bit more about how single cell, sequencing and so forth could help us better understand sex differences. I mean, one thing that came to mind as you were talking was the X chromosome silencing. And that can vary in different cell lines, which chromosome is silenced. But how else do you think that single cell technology might? Especially, there's a lot of students on the call who might not know what single cell sequence, or what the intuition behind it is, why it's important in the future. Because a lot of us are social scientists, so we don't have
So I didn't know exactly who the audience was going to be, so that's why I was like, genetic correlation, if you don't know, is blah blah blah, because I wasn't sure. But so
the background,
so I'm not an expert by any means on single cell. When I've seen single cell mentioned most in regards to sex differences, it's like you said is in regards to the X chromosome inactivation, because some cells are going to have one copy versus other cells are going to have another copy. It's not the same. So beyond that, the most important thing that I've seen is because some tissues are going to have, so if you look at bulk tissue for males and bulk tissue for females, the composition is not going to be the same. So tissue has different types of cells within it. So if there is a difference in the number of cells for a different type of cell within a tissue between males and females, that could lead you to believe that there is a difference in gene expression regulation, when truly it just means that there is a difference in a certain cell type within a particular tissue within the sexes. So that could act as a confounding factor. So that is essentially where the line of where I've seen single cell being mentioned the most in regards to this.
Is it possible though to just control for cell type proportions?
So in the latest GTEx paper, they actually found that by controlling for differences in cell type, you did lose a lot for example, for breast tissue. You lost a lot of the sex biased EQTLs for breast tissue by controlling for cell type. I think the problem that it said is it wasn't possible to know exactly the cell types, or the getting the idea of the actual composition of bulk tissue wasn't as straightforward as it seemed. So that was why you couldn't really correct for it. Again, I might have to look into that, but that I believe is what they stated, why they couldn't really do that for all tissues. They were only able to do it for several tissues.
Okay, so it would give us like a lot more precise measurement of confounding and okay, great.
So Elena, one of our postdocs, Joseph, has a chat which is essentially about thoughts on methylation as a mechanism you want to, you can take it however you want from there, but that's the key point.
Yeah, so we haven't looked at methylation data, and I think it would be extremely interesting to look at methylation data. I'm now in my postdoc starting to look at methylation data, so I'm fairly new to that world. Yeah, so I think there's definitely a lot of potential into looking at, like I said, methylome differences have been found between the sexes. We haven't seen if there's any overlap between these SD SNPs and whether they are particularly enriched in a particular epigenetic mark at all. So yeah, definitely. I mean, it's definitely something that if I had more time, I would have liked to look at, because it's another line of potential way of explaining the regulatory consequences of SD SNPs, or somehow try to explain a particular pattern that could be leading to the detection of SD SNPs. So yeah, it's worthwhile. That's why I included the multi-omics, where all of this stuff yet to be explored.
Thanks. Yushang, can you unmute and ask your question?
Hi, this is Yushang. I really like your slides. It's very beautiful. So, I have a question about participation bias, because it's well known that there's participant bias between males and females. So I wonder whether you take that into consideration when you do your analysis. Thanks.
Yeah, so for people that don't know about participation bias is when males and females participate non-randomly. So for example, in UK Biobank, you might have that very lean males are participating whilst females might be participating that for whatever reason are not that lean. So you might find an association. If you were to run a genome-wide association study on sex, you might find peaks or associations that truly don't really have anything to do with sex determining. It's just that, like I mentioned, there's not non-random participation between males and females. And there's a study that came out recently where they talk about this. And they found that these signals, if you were to run a genome-wide association study on sex, only show up in cohorts where participation occurs, like UK Biobank. If you look at cohorts where there is no participation, you wouldn't find these signals. So yes, we accounted for that by essentially running a genome-wide association study on sex, finding the SNPs that were showing up as significant and removing them. So we didn't consider that in the this comparison.
Thanks.
Yeah.
Any other questions from the group? I have one kind of general question, Elena, about I don't know what the number is, but let's say the UK Biobank has 20,000 phenotypes available. It's some larger number than 530. I'm wondering, maybe by a lot, and I'm wondering about the process of selecting the subset of the phenotypes and what you purposely discarded and what you purposely focused on. Can you say a little few words about that?
Yeah, definitely. So we started, my work is a continuation of previous work from other people in my lab with something called Gene Atlas. For Gene Atlas, we had in total 776 phenotypes, which I believe was, I think, I'm not exactly sure how they called it down, but I think it was the majority, if not all, of the phenotypes that were available at that point in time, or that we had access to. So from those 776 phenotypes, I culled it down even further. First of all, we're only looking at non-sex specific phenotypes because some of them were breast cancer, some of them were ovarian cancer. So we couldn't really compare genetic effects for males and females for that. Then we also limited, because of statistical power, we were limited to traits that had at least 400 cases in each sex. And that's to do with statistical power and the fact that case control imbalance can lead to false positives when using linear mixed models. So we used those, and there were a couple that were just, when doing our manual quality control, they seemed a bit strange. And those, I believe, I've talked to other people and they seem to have the same issues. There were some blood phenotypes in UK Biobank, like monocyte count, where if you look at the distribution, it's a bit strange. I'm not sure how accurate the measurement is. And yeah, I think in total, after all of that, which the process is defined in more detail in the paper, we ended up with 530.
Like I saw it, I'm trying to think of which one this is. The number of vehicles in a household is listed on one of the figures, but is it to say that I should assume that that, education and a lot of the social stuff is included or excluded?
Well, I didn't remove it, so I would expect it to be in there.
Okay.
I'm not exactly sure, but it should be in there. I mean, I definitely didn't remove it.
Yeah. Any other questions from the, let's see, Q?
Just want to follow up on what Jason just said. So, but the biomarkers are not in the, right?
No, the biomarkers. You
didn't do the GWAS on testosterone.
No, they weren't available yet when we ran the models.
I see.
Maybe that's an answer to that, a follow-up question of are there 10 more phenotypes you wish you would have had?
Yeah, those.
And what would those be, like testosterone and?
Yeah,
related?
testosterone, estrogen, I think hormones, definitely. I mean, there's also other studies have had access to LDL, cholesterol, all that stuff. Yeah, biomarkers, especially because biomarkers generally, the interpretation of genome-wide association studies are supposedly easier. If you look at the signals, it's technically easier to understand why they're popping up, where in the pathway, be that biogenesis, be that something else, what they actually mean. When you get a complex phenotype, you many times just don't know what each signal means. So it would be an interesting exercise.
One last, any other last questions? Otherwise, no one else raises their hand. We're going to give Elena a 15-minute break before her few one-on-one sessions, and then just join me in thanking her for presenting to our group. An excellent presentation.
Thank you.